All right, we're starting in section 1 in chapter 7. And chapter 7 deals with membrane transport and how things get across the membrane and what impact does that have on the cell. So we're going to start off understanding the structure of the membrane before we get into the function of it. So looking at the structure and breaking it down, first of all, we talked about lipids in the last chapter or so, and one of those lipids were phospholipids. So we're going to spend a little time in this chapter really focusing in on phospholipids and their role in our cells. So phospholipids are the backbone of cell membranes. They're the most abundant lipid in the plasma membrane. They have a unique characteristic about them, and we talked about that before because we said most lipids are nonpolar or don't have charges, but phospholipids have a charge because of that phosphate head that is attached to the glycerol when you look at the structure of a phospholipid. And so since it has that charge, it makes the tail region hydrophobic, right, or nonpolar or water-fearing, but it makes the head region with the phosphate group hydrophilic or water-loving or polar. And so since it has this dual personality of hydrophobic and hydrophilic areas, we call phospholipids amphipathic. Amphi meaning dual life, right? We see that in the word amphibian as well, but they have both hydrophobic and hydrophilic areas. And when we talk about the membrane, we have described the structure of the cell membrane as a fluid mosaic model, meaning that the membrane is very dynamic. It's always moving. It's a fluid-like structure, but it's a mosaic because it has a lot of different materials in it, specifically proteins, a lot of different proteins in it. And so the basic structure of our membranes are lipids and proteins doing various jobs, and there were lots of experiments done to try to figure out the structure of a membrane well before electron microscopes were available, and so we're going to look at what that was. But we did say that membranes are made out of proteins, but that phospholipid bilayer, when you look at it, that's the structure, and we can break it down, and you can see the part that is dissolved in the water. That's where you're going to find the phosphate group, and you can see the water molecules being attracted to the phosphate group, and then you can see the nonpolar tails turning in towards each other, forming that hydrophobic region on membranes. So two major experiments or pieces of research that were done to try to describe the structure of the membrane. The first one was done by Davison and Dinelli in 1935, so again, well before electron microscopes. Their first proposition was that a membrane is like a sandwich, with the phospholipid layer being sandwiched between two layers of proteins. They figured that because the phospholipid would not want to be by water, and the proteins they originally, but incorrectly assumed, were all hydrophilic. That model, that sandwich model, was disproven when they figured out that proteins are actually also amphipathic. They also have hydrophilic and hydrophobic portions as well as the phospholipids. So that sandwich model by Davison and Dinelli was tossed, and in 1972, with the help of electron microscopes and freeze fracturing, they determined that the membrane is a mosaic. So we have the phospholipids making up the majority of the structure, with the proteins dispersed within it, kind of floating around like buoys out in the ocean, and the only part of the proteins that are exposed to the aqueous extracellular matrix, which is on the outside of the cell, or the aqueous internal cellular environment, the only parts of the proteins that would be exposed to those two watery environments would be the hydrophilic regions. The proteins actually had hydrophobic cores that are found spanning the phospholipid bilayer. So 1972 is the year that we developed this current model that we call the fluid mosaic model. So you can see some proteins being attached only to the outside, We call those peripheral. And then you can see other proteins that span the whole membrane. Those obviously have to have hydrophilic regions on the outside and hydrophobic regions on the inside. So again, they did freeze fracture membranes, and those freeze fracture studies basically split the membrane into two halves. And so what they did with this technique is a splitting along the membrane's bilayer and basically opening it up so they could see what's on the inside of a membrane. Because you can see what's on either side of the membrane by looking at it with an electron microscope, but to really understand, and you can chemically deduce what's in a bilayer as well by taking it apart, but you want to see how it's built. So in order to see what's on the inside of the membrane, they did freeze fracturing, which basically is freezing a membrane, or in this case they're showing you freezing it with, and this is obviously a simplified model, but freezing a cell, and then with a microscopic knife taking that cell and shearing off the outer layer of the membrane, in effect opening up the membrane so you can see how the proteins embed themselves in the membrane, and you can see the two different layers. And so when you see the pictures on the on the right, you can see inside the extracellular layer and you can see inside of the cytoplasmic layer. So you can see that there's definitely two different looks to those views of our layers there. So how do we maintain fluidity? Well, because we said it's a fluid mosaic model. So phospholipids can move about within the plasma membrane. They move around, they drift, as well as do some proteins. Some proteins are kind of anchored where they're supposed to be, but some also drift about, and they drift about laterally. So if you are on the extracellular matrix side, right, the outside of the cell, then you'll stay on the outside of the cell. If you're a phospholipid on the inside or the cytoplasmic side, then you'll stay on that side. Very, very rarely does a phospholipid or a molecule actually switch from one side to the other or flip-flop transversely across the membrane. And that makes sense because, you know, if these proteins or if these phospholipids, since they have hydrophilic and hydrophobic regions, if you had to cross over the membrane, then the hydrophilic side would have to cross through the hydrophobic core, and that doesn't work very well, right? In that case, opposites do not attract. We know like dissolves like. So those things with a charge, those hydrophilic regions can't quite get through. So when you look at membrane movement up there at the top, lateral movement, moving a lot, 10 to the 7 times per second. That's very quickly, a lot of movement laterally. But once per month will a molecule flip-flop across the membrane. So it obviously is not a very common process. Then we also want to talk about, you know, how do we maintain this fluidity as temperatures change? Because we have to maintain homeostasis. So our cells, which are made up of and surrounded by cell membranes, which are fluid oil to a certain extent, how do we maintain that structure if the temperature changes? So as the temperature gets more cools down, right? As your body temperature cools, as the external environment starts to cool, then the membranes go from a more fluid state to a more solid state, which makes them more stable, I suppose. But you know, think about cooking oil. When it goes from an oil to a solid, like maybe if it's in the refrigerator, things can't get through there. And so we need to have, we need to maintain that fluid nature of the membrane. We don't want it to become solid or the cell will die because it can't allow stuff across it. So the temperature that the membrane solidifies at is really dependent upon what lipid makes it up. Okay, so if you are living in a cooler environment, if you are, you know, an Arctic fox, or if you're winter wheat, or something that lives when or where it's cold, you will have more unsaturated fatty acids in your membranes, because remember, unsaturated fatty acids are liquid at room temperature and will stay liquid longer than saturated fats. Saturated fatty acids turn solid more quickly than unsaturated fats do. And we need our membranes to be fluid. That's how they work the best. That's how things can get through them that need to get through them with this movement. And if they are not fluid, they don't move, and we have a problem. And so when you look at my two membranes there, right, a fluid membrane has a lot of unsaturated fatty acids with the kinks in the tails, so those phospholipids stay spread out and stuff can move through it. On the other hand, if you have a membrane with a lot of saturated fatty acids, you can see how those hydrocarbon tails pack in real tight, making the membrane more viscous, more thick, less movement. Another way that we can help maintain fluidity and stability of a membrane is by the steroid cholesterol. Cholesterol is an important steroid. We talked about it before. Too much of it's bad, obviously, but in your membrane it's important because it has different effects. It can do different things depending upon the temperature of the organism. So as temperatures start to warm up, obviously fatty acids and oils and lipids will get more and more fluid. They will move around more. So what cholesterol does at those warmer temperatures is it kind of acts like scaffolding, like a stability, like an anchor, and it restrains the movement of the phospholipids, keeping them in place, or at least better in place than they would be if they didn't have cholesterol there. So cholesterol can help resist movement of the phospholipids as the temperature gets warmer. Now, on the other hand, as the temperature gets cooler and that membrane wants to turn solid, it can maintain fluidity because it prevents those phospholipids from getting really tightly packed. So those cholesterols act as spacers in a membrane that's trying to become solid as it gets cooler. And so that space is important because it can continue to allow stuff to get through and it will space out the phospholipids. Right, so there we see cholesterol in the membrane. Again, if the membrane gets too fluid, the cholesterol kind of stabilizes the structure. If it starts to get too solid, you can see how it would provide openings and some pores through the membrane. Okay, so that takes us to another aspect of this mosaic, right? Phospholipids, cholesterol, all lend to the mosaic idea of a membrane, but so do proteins, and there's lots of different proteins that are embedded within the lipid bilayer. Proteins are what make membranes do their job, and they are specific to the cells that they're in. So certain proteins will always be found on certain cells. You can see by this picture, there's lots of different types of proteins with lots of different extensions, doing lots of different jobs. But we really have two basic types of proteins that you can identify on a membrane. Now those basic types will have different jobs and different names, but we have peripheral proteins, and those are bound to the surface of the membrane only. So peripheral, like our peripheral vision is out to the side, these proteins are bound to the sides or the surface of the membrane. Whereas integral proteins, these guys are integrated right into the membrane, and they go through the core of the membrane. They penetrate that hydrophobic core and can work their way all the way through the membrane. And when those integral proteins do manage to span the membrane, and they do go from one side of the membrane to the other, that's when we refer to them as transmembrane proteins, because they can span the membrane, they can get across the membrane. And in order to do that, your integral proteins must have both hydrophilic regions on the sides or on the ends and a hydrophobic region in the core, right? So those hydrophobic regions on the protein is usually composed of your nonpolar amino acids that have, the R groups that make them nonpolar. And so here's just an example of a transmembrane protein. You can see the N terminus of my amino acid, the C terminus of the amino acid, and how it's coiled up into that helical state, keeping the hydrophobic core in the middle of the protein and then the hydrophilic regions out on the sides. So proteins will do 6 big jobs in a cell. There are 6 different things they can do. They can transport material across the cell. They can be involved in enzymatic activity, encouraging reactions to happen as they are built into the membrane. They are involved in signal transduction, which means they can receive messages or molecules from the outside of the cell and send that message to the inside of the cell. This is how cells communicate. Cell recognition, they can be part of a network of proteins that help other cells identify each other and then bond together with various ligands or carbohydrates or lipids attached to it. Intracellular joining, they can be part of an anchor system that help hold cells together, and they're also used to attach both to the cytoskeleton and then to the fibers of the ECM. So they can be anchors holding that cell in place to help form tissue. And so here we see the different jobs: transporting materials across the cell, converting products to reactants in enzymatic activity, being a signal transduction molecule, right, receiving the signal on one side of the membrane and then sending the signal on into the cell. Then you can see cell-cell recognition. You have one protein that might have an attachment, maybe a carbohydrate that is then recognized by another protein on another cell, or maybe they're used as joints in the holding the cells together, or as attachment points holding the cell to the ECM and the cytoskeleton. And then we also need to know that carbohydrates are important in recognition of cells. So cells can't talk, they can't read, they can't see, right? They have to recognize each other by binding of of signals to certain receptors on the cell membrane. So these surface molecules, which are typically carbohydrates, are on the plasma membrane, and they act like flags. They act like ways to recognize cells, between cells. So these membrane carbohydrates are going to be bonded to lipids on the phospholipid membrane. Then we call them glycolipids, or the carbohydrates might be bonded to proteins, forming a glycoprotein. And this is a very specific marking system. So these carbohydrates on the outside of a cell, right, on that ECM side of the cell, varies greatly among species, individuals, types of cells. I mean, it's very, very specific as to what carbohydrate is found on the outside of the cell. It's a unique marking system, unique to a specific cell. And then lastly, membranes also have very distinct inside and outside faces. The outside of the cell will look different than the inside of the cell, and that's a little tricky because we have to make them that way. They don't just happen that way. So this distribution of proteins, which is not equal on both sides, you might have more proteins on the outside. You'll have more glycolipids or glycoproteins on the outside than you will on the inside. So how does that happen? Well, it happens through the production of these membranes through the endomembrane system. And that endomembrane system orients the inside and outside of the vacuoles that they are producing, because these vacuoles just become part of the membrane. And so they are definitely organized in a way to line up and get what's supposed to be on the outside to the outside and what's supposed to be to the inside on the inside. And so you can see this image here as they flip-flop, as they move from the ER to the Golgi to the plasma membrane, you get a sidedness to them.